Hello. I hope you can hear me all right. I would not be able to turn on my camera today. I don't have a great internet connection. So we'll just start like that. Can one of you confirm if you can hear me all right? And I'll start sharing my screen in the meantime. Yeah, okay, perfect. Thank you, Hema. Alrighty then. Welcome to the seventh live session of the NPTEL course, Cell Culture Technologies. And those of you here, you've been joining us for almost all the sessions, so I'll skip through all of this. So yeah, we are through with three-fourth of the course. This is the penultimate session, penultimate week, and we have one more to go after today. And as always, the session is being recorded, and this recording will be there on YouTube on this channel. And any material we discuss today, which is primarily this PPT, that's going to be uploaded on the Google Drive. And before we start, are there any questions? Any questions about any of the previous weeks or any general questions not exactly pertaining to this lecture? Or even if they're pertaining to this week, are there any particular questions or any things that you would like me to talk about? So I'm seeing a few new names today, so welcome everybody. Um, Yeah, no questions. All right, that's great. So, for those of you who might be joining this live session for the first time, throughout the session, please feel free to drop in your questions, either using the chat feature or by simply unmuting your mic, and yeah. So I am not able to turn on my camera today, so it might look a bit weird to you, but yeah, I don't have great internet connection today. So yeah, if there's nothing from your end, then I'll just start the session. Maybe today's session may not be very long, unless you have a lot of questions. But anyway, let's dive into it. All right. So this is where we were last week, where two cell separation techniques were discussed, and I'll just quickly revise that. So, why would you want to separate cells out? So, whenever you want to culture cells, more often than not, in a majority of cases, people are interested in culturing one type of cell at a time. Now, where do you get your cell from that you want to culture? So, of course, you can buy cells. There are cell lines which are available where people have already purified and they are just ready to culture. But in a lot of cases, what people want to do is they want to take cells directly from an animal. That could, so in most cases, that's either mouse or rat. There could also be human cells that are taken from subjects. So, there could be biopsies um, that are done or samples which are taken just to check a few things. So, in either case, when you want to take cells directly from an animal source, human, non human alike, you more often than not will not get that single type of cell purely. You will get a mix of multiple cells. Now, if you want to culture all of them together, then it's great. You don't need to do this step. But if you want to start off with um, if you just want one or two or particular type or subtype of cells from that group of cells that you get from the animal, then in that case you need to separate out cells. So first thing is you dissect out the tissue or organ of interest, and then it is dissociated into single cell suspension. So we've talked extensively about this in the last week or two, where you can use enzymatic um, dissociation, you can use mechanical dissociation to make a single cell suspension. So cell cell contacts are broken, and so are the cell ECM contacts. So you just have pure cells with you in some sort of a medium. So when you want to separate those cells out, if the different types of cells have different densities, then um, if you have, so again, it's going to be a very simple drawing, but if this is a tube that you take, generally we talk about these conical, like they're conical from the bottom. And if you have different layers, suppose this is, you have different densities, D1, D2, D3, like that. And if you add your cells here at the top, and then some of them will settle here, some will settle here, and some are going to settle here, depending on their densities, then you'll say that, okay, you can separate out whatever cells came here, whatever cells came here, and whatever came here. Now, if the three different types of cells you got, if your cell suspension had, suppose, three different types and they got into three different layers, then it's great. Your job is done. You've separated out the cells. Of course, there's a few optimizations and checks you would want to do, but that's irrespective of any method you use. But now, what if you had two types of cells which have very similar densities? So no matter how many layers you make, you will not be able to separate those two types of cells perfectly well, right? So in that case, you need to go to some other method. So density gradient centrifugation. So density-based separation will not work. In that case, we go on to something known as markers, which are cell type specific. Now, of course, when we say markers, so some sort of, so we'll build on this one by one. So a lot of proteins are expressed inside the cells and on the receptor of the on the surface of the cell, which is on the membrane, and a lot of proteins are secreted out of the cells depending on what sort of cell it is. Now, secretory proteins are not going to be too helpful for us because they'll be outside the cell, they'll be out and away from the cell. So two things that we can use are proteins or some unique molecule which is either inside the cell or which is expressed on the surface. So we'll start off by talking about these cell surface proteins, which are generally receptors. And then we'll see why some of these things can also be done when the protein is inside the cell. Okay. So two things. So the one thing that we discussed last week was immunopanning. I don't think we explicitly called it immunopanning last week, but that's what the thing is. So what you'll do is you have your culture dish, you add your antibodies. So again, antibodies are drawn with some sort of a Y shape. So you have these antibodies that are attached onto your cell surface. Then you add your cell suspension here. Okay, again, the mixed cell single cell suspension you add here. Some cells are going to attach to this antibody because they have some receptors, and some of the cells will not. Now, depending on which one you want to collect, you can collect that. Okay. So cells, so normally what's going to happen is, okay, I have these four types of cells in my single cell suspension. I want to collect one of them, which expresses a protein A, and now I have the antibody against that protein A. So remember, this protein here will be known as the antigen. I'm just calling it AG for now. And this thing is your antibody. So antibodies are against a particular antigen. And normally all of these are proteins. All right. So of course, the specificity of what you collect depends on when is this marker expressed in your cell. Is it expressed at a particular time point, at a particular developmental stage? Or do multiple developmental stages, so suppose we take the example of neurons, we've talked about a lot of embryonic stage and fetal stage neurons so far in the last 2-3 weeks. So for example, if you want to look at neurons from, let's say, you were saying, okay, neuron expresses something called beta 3 tubulin. Okay, do neurons express beta 3 tubulin at E14? Do they start expressing it at E18, or do they only express it after the mice is born? Because yes, it's a fact that beta 3 tubulin is a neuronal marker, but is it a marker of um, just of all neurons? Is it a marker of a particular subtype of neurons? Is it a marker that starts from the very start of neurons themselves, or do only mature neurons express? So something like that. So you need to know that. And then, are they expressed in all neurons? Are they expressed in spinal cord neurons? Are they there in the peripheral neurons, interneurons, hippocampal, non-hippocampal, and so on, right? So anyway, so your uh, this immuno-based precipitation, not precipitation, sorry, immuno-based separation, that is a great technique because antibody antigen are super super specific. One antibody ideally only recognizes the antigen that it was raised against. So antibodies are made by your B cells. That's happening in your body also. So when you are given a vaccine, you are given a small dose of a protein or a small molecule that is normally a part of that infectious agent. So, for example, for a virus or a bacteria, there are things which are expressed on that virus or bacteria's surface. That thing alone, and not the entire virus, that thing alone in different forms, either as RNA or as protein, that is given to you in the form of vaccine. Your body say, okay, that's a foreign object. Now the body is not harmed by it because it's not the actual virus, but the body can still recognize it as a foreign entity. So it says that, okay, my B cells are now going to work and they're going to make an antibody against that antigen. And now the B cells, they also remember it. Again, that's more immuno, so we won't go into it, but your B cells can remember that. And then when the actual infection, if it happens, then the body is already prepared. So it does not lose time in starting that process of making antibodies. It can just immediately recognize and increase that production. Okay. And that's how antibodies are made in other organisms also. So mice, rat, chicken, donkey, these are very commonly used to make antibodies. All right. So, okay, these are just side discussions which are not exactly what was discussed in the lectures, but I hope this helps. So, if you think this is too much of a digression, then just let me know. But yeah, I think this is going to be a little bit helpful. Okay, so that's how immunoseparation, immunopanning is done. Now, another way, so of course, this is a little bit cumbersome because you have your cell suspension, you want to add it, you want to coat your, you want to coat the surface with antibodies, remove something, keep something. It's a lot of manual work. And of course, if you want to separate two different cell types, four different cell types, it is difficult, right? And then, yeah, so even downstream of that can be a little bit um, tedious. So there's a smoother way, more efficient way of doing this. So we'll talk about two terms which are related, but they're not the same. So one is flow cytometry and the other is fluorescence associated cell sorting. Okay. So the principle is um, the same. So what happens is if you have an instrument where your cells, so you already, again, in all of these things, your starting material is a single cell suspension. So if you have an instrument, so we'll just come to this. So we're calling it facts for now, but the principle is quite similar up till the first part for your flow cytometry also. So in this case, what you do is you have your single cells. Now, in this, so it's just a diagram I've taken from the internet, but this is a single cell suspension. Okay, so these are separated out. These are not clumps. They're separated out. So what you do, again, the same thing. You add fluorescent antibodies onto this. In the previous case, the fluorescent sorry, not fluorescence, the antibodies were there on your culture dish. But now in this case, your antibodies, forget about fluorescence for a second, your antibodies are added to your cells, and it's like either the cell will attach to an antibody or the antibody attaches to the cell. It's the same principle. It's just that in the previous case, the antibodies were attached to a surface, to your well or to the plate, so they couldn't move. So now that they've captured the cell also can't move. But now in this case, nothing is stuck to anything. So your antibodies, of course, antibodies are really tiny. Your cells are huge, right? So antibodies are just, they have labeled the cells. They have now covered the cells. Okay. So in this case, you can see that antibody now has a fluorescent tag on it. So we won't go into the specifics, but you can, let's say your antibody now is, so again, this is a molecule. Even though we have drawn it like a Y shape, it's a molecule which has some free ends here and there. Okay, so in that case, you can tag or you can covalently bind a dye or a fluorophore onto this. And whatever you bind it, so typically you have things which are red in color, green in color, blue in color. We've seen some fluorescence images in the past few weeks. You can just Google and say what immunofluorescence looks like. So your antibodies now, so wherever the antibodies go, you'll see some sort of a color. So in this case, you can see that the antibodies have a purple color on them. And these antibodies are specific to just the purple cells that are marked here. And you can see that there's a slight glow that you can see around these purple cells. So up until the previous point, up until here, they're not glowing, but now some cells have been labeled. So cell labeling has been done. Some cells are now glowing purple. Okay. Now you load the cells. This labeled cells, labeled, unlabeled, everything, all the cells, because at this point you've not separated out, but you know some cells are going to be labeled, some are not labeled. You load them onto a machine. Now, depending on what can the machine do, you either classify that into FACS or flow cytometry. Okay, so the first step is that the cell suspension is flowing through. That's the flow word in flow cytometry. So what this does is, again, through some sort of a nozzle, which is allowing only one cell to pass at a time, it's making droplets, which are like really tiny, very little amount of the liquid. So obviously these cells are in some medium, some liquid. Okay, whatever is allowing these cells to at least stay happy for whatever time this entire process is going to take. So it makes droplets. Typically, the that droplet, like it's a few nanoliters along with each cell. So maybe like 3 to 5 nanoliters around each cell. That's the size of the droplet that we're talking about. So nanoliters is 10 to the power 9 liters. Okay. Normally you talk about milliliters and microliters in the lab. This is nanoliters. So 10 to the power minus 3. Okay. So it makes this, it makes a some sort of a droplet, and the size of the nozzle is such that only one cell, as you can see. So again, this is just a schematic diagram, but mostly what it does is just one cell at a time can go through. It can be error prone, but we'll assume for now that only one cell can go at a time. And then there is some sort of a laser that falls onto the cells one at a time again as it's flowing out of this droplet generator. And there are multiple detectors. Now, depending on what detectors there are, um, mostly you are going to see two sorts of things. One is known as a forward scatter, one is known as a side scatter. These are your non-fluorescent detectors. So what the forward scatter tells you. So basically, some light falls on the cell, some goes through, some gets scattered here and there. So whatever is going through, that forward scatter is telling you how big the cell is. So the forward detector generally tells you the size. If it's small, is it big, and so on. The side scatter tells you the complexity or the granularity within the cell. So some cells are going to be very plain, some cells have a lot of complex things going on. Again, it's just telling you about the, if you can think about it, the first one is the size, and the second one is its internal features. And what, so this is an analyzer, of course, so something's being analyzed, and this is the sort of plot you would see. So again, this is not super important from the context of your course here because it was not discussed in too much detail. But since we do have some time today, I think the lecture, today's class would be a little bit smaller. So I just thought of including this. So you can see here, it's a 2D scatter plot. Each dot that you see here is a cell that was analyzed. And if I remember correctly, these would be something around 10,000 cells that were looked at. So you can see here that there's some sort of a distribution along the x-axis. So the bigger the cell, the more complex it is. But not always. And then you can see in this case, some cells were selected. So again, this is what is everything. Now we think, okay, the cells which are too small and they don't have as much, I would say, granularity, they're probably dead cells. Also, cells which, it's the increase is not in the same ratio. So again, this is from experience. This is how a lot of people, so this entire thing was done by an operator of facts. I did not do this myself, but from their experience they could tell that generally cells which are lying below that straight line would be mostly what you would be interested in. These would be good cells. These would be the cells that you like. Now this was a cell suspension of the same cell. These were all stem cells. So here you don't see different populations, but your graph, this plot would look roughly the same. It's just that smaller size cells would be on the left versus on the right of, so this is forward scatter. If you see, FSC stands for forward scatter and SSC stands for side scatter. So forward scatter is for your size, side scatter is for your complexity. All right, coming back, if this is all that your instrument can do, then we call it flow cytometry. So cytometry means, cyto means cell, metry means some sort of metrics or calculations or values. So if you are quantifying what your cells look like, basically what they're looking like, because this is all, based on their scattering properties, then we call it flow cytometry. Flow cytometry. But now, if the instrument can go one step ahead, and in addition to that, it can also separate out your cells. Now, separating out cells can be done based on both your size and complexity that we just saw. Plus, it can do one more thing. Now, in addition to just looking at it, even for flow cytometry, what it can do is it can also look at which of your cells are fluorescently labeled. Again, that can also be done in flow cytometry. So yeah. Okay, so yes, so your flow cytometry and FACS both, they can look at, again, the size, shape, complexity. And also what you can see here is, so again, this is just some more filtering based on more scatters. But you look at this plot and then we look at this plot. So what you can see here, again, the x-axis is the same, the forward scatter, which is size. And then once we've selected this population, if you look at how many of them are expressing GFP, so GFP stands for a green fluorescent protein. So you can see that, okay, if you put some sort of a threshold, you're looking at just, the very, very highly green expressing cells. So you can see that there's some population which is GFP positive and a large population, as you can see, there's a lot of dots here, that it's GFP negative. Okay. Again, these are some metrics that you might see, that if you have 100% cells, how many did you find were GFP positive? So out of the total cells, so here you can see, yeah, these were 20,000 events. Out of 20,000 events, about 5%, which is about 1,000 cells, those are GFP positive. Okay, so again, this first bit, so based on your forward scatter, side scatter, fluorescence, all of that, if you're doing, that's flow cytometry. So based on your flow, you are quantifying some features, size, shape, granularity, fluorescence. Now the second step, if it can do cell sorting, and normally people do cell sorting based on fluorescence. Granularity, size, shape, these are not enough to sort cells out. As you can imagine, if these were enough, probably even your these density gradients would have been enough. But what people want to do is based on fluorescence. So the name of the instrument is FACS, but you can also do cell sorting just based on your size and shape. Okay, so what gets done next is, depending on how complex the machine is and how many types of cells it can separate out at the same time. So in this case, what we did was, we had some cells which are fluorescent, some cells which were not. So what we do is, we put a positive charge on some cells. Let's say we put a positive charge on the cells we want to collect. And then there's a plate which is negatively charged. So our positively charged cells are going to get attracted to this negative plate and they can be collected. Let's suppose there was another fluorescent antibody which was blue in color, and then you could apply a negative charge onto those blue colored cells, and then they'll be attracted to the positive plate and then be collected in a separate cell collecting tube at the same time. And whatever were not charged in either type, then they can just be collected, and they can be like an extra cell, like for the undeflected droplets, for the ones that you don't want to collect, right? So mostly it's so again, there's a lot of complexity that can be introduced. So the detectors can be multiple at the same time. You can have detectors for, again, like I said, the four primary colors that people use: blue, green, red, and orange. Um, so you can have multiple graphs. You can say that, okay, I want cells which have blue and orange, but not red, things like that. Collecting at a time generally is just one or two. As you can see, you can only have some charge being put. So at this point, this is where the bottleneck is. But depending on your detectors, you can detect ideally all colors at the same time, right? Are we good so far? Do you have any questions about facts and flow cytometry? Yeah, just give me a thumbs up if everything is all right. Okay, all right, great. Thank you so much. Just give me a second. Oh, yeah. So, okay. So next up, again, this is just a little bit more recap from the last week. So this week, what we're going to be talking about, so what was introduced a little bit was, um, so for example, oh yeah, so this was as a use case of fact sorting. So if I tell you that there are multiple types of motor neurons in the spinal cord, these are called spinal motor neurons, or SPMN. So MN is a very common acronym used for motor neurons, and SP stands for spinal motor neurons. Primarily, there are two subtypes, alpha and gamma. The beta subtypes are very, very uncommon, so we'll not talk about that. So, for example, if it is said that the alpha subtype of neurons, alpha subtype of spinal motor neurons, they express something called the HB9. Okay, that's a gene, and the protein product is expressed in just the alpha subtype, and the gamma subtype do not express it. So if you want to isolate the alpha motor neurons, then you can have an antibody against this. Now, coming to why you should be careful with this statement is, you need to be sure that, okay, is it expressed in alpha motor neurons at all developmental time points? Can I use an E18 mice? Can I use a postnatal mice for this? Can I use a, or mice or rat, whatever the organism is? Can I use either of them? Or can I use an E8 for that? So the question is, you are only interested in alpha motor neurons. If you just want to isolate alpha motor neurons. So the thing is that around E, something around E14 is when the alpha motor neurons strongly express this marker, and then slowly this is expressed by other cell types too. So if you have a mice, an embryo, a mice embryo which is younger than E14, and you use this thing to um, purify your cell, then it will not work because these cells were not expressing HB9 at all. Versus if you talk about a later stage of mice, maybe E22 or a postnatal mice, then in that case, this may not be specific to just the alpha motor neurons and may be expressed by more types of neurons or motor neurons. All right. So again, just as a reminder, it's just good to know because this is probably new for most of you. So This is what a cross section of the spinal cord looks like. Um, you have cells which are, you have neurons which are entering the spinal cord, which are bringing back some sensory information from your limbs. That's the input, that's the sensory, and that's coming from the dorsal side. Okay, so that's the back. So this is your dorsal side, this is your ventral side, this is going to be the right, and this is going to be the left. Okay, so dorsal is back. So as you can see here, the input is coming from the back, it's getting processed, and the output is going from the front. Ventral is front. So so this is where we had talked about the dorsal root ganglion last week. So all the nerves, their cell bodies, which is where your nucleus is, all the neurons, their cell bodies are together at one place right before entering the spinal cord. So that is a place where you'll have a lot of neurons if you want to isolate them. So that's called the dorsal root ganglion, and that's outside the spinal cord, so as to say. And if you talk about what's inside the spinal cord, you have things which look like some sort of a butterfly shape. Again, the part which is towards the back, that's called the dorsal horn. What is towards the front is the ventral horn, and whatever's on the sides is the lateral horn. Okay, so again, that's just a couple of good diagrams to help you visualize what we're talking about. One more thing which I forgot to talk about. So yeah, so in this case, we talked about so these antibodies, they were against a protein or something, some antigen, which was outside the cell. Now, what if you are not able to identify something that is outside? What if you are able to, you say, okay, there's a very specific marker for my cells, but that is expressed inside the cell. So it stays inside, it's never expressed on the surface. What do you think you can do about that? It's an open question. There could be a couple of things that can be done. So the idea is that you have a cell. If the cell expresses something on the outside, then what you can do is, suppose you have this GFP that was here, we talked about this GFP here. So what you can do is, you can simply have your antibody outside, right? You can just have this here and your cells are being labeled. But what if there was another case that there's the cell, but whatever it was expressing was inside? How do you label something that's on the inside of the cell? Right? This first step, cell labeling, how do you do that? Can you do that? Can you do facts on a cell? Like, can you sort out a cell through facts when you know the marker is inside the cell? So, any idea? Would any of you like to discuss? It's an open question. So, I just want to learn. Like, I just want to understand what your thinking is, and it's okay if it's wrong. Absolutely okay if it's wrong. So you can chat. You can unmute yourself. Unmuting would be quick if you can. So yeah, anybody? Okay, all right. So there are a couple of things that you can do. So of course, one of the things is, so one of the easier ways is actually to make something like a transgenic system where intrinsically your cells, whenever they express this protein, they also express a fluorescent tag with them. You're no longer dependent on an antibody now. If already this, so instead of the green antibody, you already have this protein being green itself. Okay. So again, this is not always possible, but if intrinsically you can have this protein, this molecule itself being green or any other color. So what is normally done is, if this is the protein, Then after that, you just add this tag in the gene itself. So you are making a transgene, you're adding some extra things in the gene which are making this green color. So it's generally GFP, but it could be other colors also. Okay, so anyway, this is just extra information. So all I wanted to say here was that in immunopanning, you are going to be restricted by receptors that are outside. In facts, it is possible that you can also sort cells which are intrinsically green, right? Because they are expressing a marker. They are only the cells that you want are green, the others are not green. But of course, that cannot always be done. If you are directly using something from a mice, then unless the mice was expressing that green thing, then you can't use this feature. So in most cases, 90% of the cases, or I don't know, 95% of the cases, you will use something which is a cell surface marker. But yeah, the other way is also possible. All right. So that was that. And we saw the use case that if you are interested in a particular cell type, and you know that at the time point that you're interested in, it has some sort of a unique marker, then you can use that. For example, here HB9 was used. Okay, moving on. Um, we move on to a little bit further from neurons and we talk about muscle cells now. So again, if you are doing in vitro differentiation from stem cells, so stem cells are pluripotent, they can become any type of cell. There are also stem cells which are specific for a particular tissue. So if you just say stem cells, may not be clear that what is the pluripotency, what is the capability of these cells to become. So what type of cell can they become? So a lot of tissues, a lot of organs in adult organisms, they have stem cells, they have their own stem cells. So there are very few, but there are stem cells in the skin. There are few, but there are stem cells in your intestine, liver, etc. Okay, so these stem cells may not be able to become a cell of the other tissue, but they do have some potency. So they can be multipotent, they can be unipotent, maybe they become just one type, but they're still stem cells, so they need to undergo a process called differentiation. Now, depending on so once you do that, there are two things that you would want to. So once you differentiate a stem cell in vitro, there are two things that you want to ascertain. One is, since they are being differentiated in vitro, so the thing is, okay, you have stem cells in culture and you have stem cells inside the cell. So are they behaving any differently? So there is a difference simply because the cells are now not in their original environment. Okay, whether these are stem cells, whether these are differentiated cells, that is one source of difference. The other source is that you're differentiating them in vitro. You're not just growing a particular cell type. You started with something and you're ending with something else. So there's a whole process that the cells undergone, that the cells underwent, right? So these are two factors, these are two aspects that can change why a cell looks different, or if a cell doesn't look different from what it should look like, then that's great, right? So suppose again, like we saw the case of neurons. So in that case, had we took neurons from, let's say, the E14 or E18 as some embryonic hippocampus, and then we cultured them. So in that case, we were not exactly doing differentiation, right? Because these were still neurons, these were already neurons to begin with. So in that case, the only difference was growing them in vitro versus them growing in vivo. But then, as we'll see some cases today, we start off with stem cells. Now, these are not pluripotent stem cells. These are stem cells that you derive from that tissue itself. And then you grow them and then you differentiate them. Okay. So again, irrespective of whatever the cell you started with and whatever you are finally wanting to achieve, two things that you want to remain consistent is phenotype. So neurons, do they grow axons and dendrites? And the second thing is functionality. Do they make Functional synapses are the neurons connecting with each other, are the neurons firing an action potential that would then um, um, be given to the next neuron through these four functional synapses. So both phenotype and functionality needs to be retained. So for example, beta cells, which are there in the pancreas, which produce insulin. Now they need to cluster together. They don't grow separately. They want to make groups. That's one of their phenotypes. And insulin production is obviously their functionality. And then today we'll talk about, for the next 15-20 minutes, or oops, sorry, half an hour, we'll talk about two types of muscle cells. So muscle cells broadly are three types: skeletal muscles, cardiac muscles, and smooth muscles. We'll talk about the first two today. And what I have in bracket are names of cell lines of these tissues. So you can take a look at them, but again, this is just for your information. So for a skeletal muscle, what you would want to look at in terms of the phenotype is something known as a myofiber assembly, and we'll walk through that in a little minute. And in terms of their functionality, um, they need to be able to contract and extend themselves. They need to have, we will see what SR is, and we'll have a little calcium spike and some, again, contraction, extension, and whatever is happening, whatever the muscles are supposed to be doing. Cardiomyocytes, which are cardiac muscles, again, their morphology, they look a different type. And then they also need to be beating. There has to be a calcium wave. So again, calcium spike, calcium wave, it's different. They're both dependent on calcium, but in skeletal muscles, it's a very small peak like that. And for cardiomyocytes, it's a more smooth, it's a wave. Okay. All right, we'll talk about that. Um, okay, so starting very quickly about skeletal muscle development. So you have these muscle stem cells. Um, these can be, you'll have different names, myoblasts and so on, also called stem cells. I am not 100% aware of what is the distinction between satellite cells and myoblasts, but if I remember correctly, satellite cells are there even in your muscles at this point, but they're not going to differentiate anytime soon unless they get activated. And once they get activated, I think that is, yeah, that's somewhere where they are called myoblasts now. So they are now back into their groove and now they're going to divide. So normally, so if you remember, we had talked about the cell cycle where you have the G1 phase and G2 phase, and there's also a G0 phase, which is called quiescent cells. They're not doing anything, they're just staying as it is. So that's the phase where your satellite cells were. And then they got activated, and they're like, okay, now we need to proliferate. We're now entering the cell cycle, we're going through mitosis. That's what proliferation is. They're called myoblasts now. And then slowly they're going to start differentiating. And so normally one cell, one nucleus, right? You can distinctly see the cell boundaries. You can distinctly see, okay, it has a darker nucleus whenever you're seeing it under the microscope. But then soon what happens is these cells, they, once they've divided, they fuse and they lose their individual identities. So now it becomes a bigger structure with multiple nuclei. That's called a myotube. And there's a lot more things happening, and that becomes a myofiber, and that gets assembled into your final muscle. So satellite cells, if you remember from the last week, satellite cells are also is also a name of so astrocytes. They're doing some supporting and clearing up these ions in your astrocytes are in the CNS. And there's a contemporary of astrocytes, just like we talked about oligodendrocytes and Schwann cells, oligodendrocytes in CNS, Schwann cells in PNS. Likewise, you have astrocytes in CNS and something known as satellite cells in the PNS, but those are different satellite cells. So if you want, you can call these myosatellite cells, but they are different. Okay, these are stem cells. Those in the PNS, those astrocyte-like cells in the PNS, they are not stem cells. Okay, all right. So these myosatellite cells are these stem cells now. Now they are not pluripotent. So these cells, they cannot become stems. They cannot become a neuron directly. They cannot become a heart muscle cell directly. But they can directly become a muscle, the skeletal muscle. So now the protocol that on yeah, so this is what is happening. So you can broadly see that there is a distinction here. One is proliferation, growth, and the other is now differentiation and actual. So all of these steps, differentiation, fusion, and maturation, this you don't need this to happen immediately. You can't do this when you just have one or two stem cells. The first step has to be its proliferation. They need to divide, right? More than division, they need to get activated and divide, right? And then does everything else happen? So we broadly classify this into two categories. So one is division and the other is differentiation. So in the body, of course, you can see here, it's just a figure taken somewhere. You don't need to remember any of these. But you can see that there are a lot of things which are ending in the term GF. This GF stands for growth factors. Now, growth factors have multiple function. Okay. Um, so growth factors, irrespective of like growth factors does not only mean literal growth. So in both cases, these are growth. Okay. So growth can be two things. It can again be division and it can be maturation or differentiation. So some growth factors like HGF, FGF2, IGF1, these are for activation and proliferation. And then you can see, okay, IGF1 is also doing proliferation, also doing differentiation. But you see here that these two terms that we saw earlier, these two terms here, right? Now, these terms here, they're actually inhibiting. This symbol is inhibition of differentiation. So initially you want this, but later on you don't want these signals because this would prevent differentiation, right? So um, this is what cell signaling, there's often a course around cell signaling in some um, degrees. So this is what signaling, this is how signaling works inside cells. You have some factors, arrow here indicates that it's promoting that step, and this P sort of a symbol, this horizontal line instead of the arrow, indicates that these factors would prevent that step. So their absence is what you want. So initially you want HGF and FGF2. Later on you don't want that. So we assume that in the blood, different blood from different animals, not necessarily human, not necessarily that organism, but you might have heard about. So in blood, if you remove the cells, then what remains is called the serum. So you might have heard about fetal bovine serum, horse serum, yeah, something like that. So those serum, they have these growth factors which allow growth, which allow proliferation. So that's the first thing. And then, uh, but of course, if you take it from an animal today versus you take it from the animal tomorrow, it will not be exactly consistent. They will be, they would have roughly the same things. They would probably have all the things, but their concentrations may not always be the same. Okay. So it's that composition, it's not a defined media, right? You don't know that you added 10 microliters of that and it's 10 nanogram per liter concentration of that and so on. You know that, okay, it's somewhere between 10 and 20. The other thing is somewhere between 5 and 10 and so on. So anyway, but that serum, so in blood, or the more accurate term is serum actually here. So serum is the non-cell part of blood. So the division phase needs the growth factors that are present in blood, but the exact composition is not consistent, versus the second part, which is your differentiation and fusion and maturation part. You don't need those growth factors anymore, right? You don't need this HGF and FGF2 anymore. You need specific cues, which are some other differentiation factors. So then in that case, the serum is no longer needed. So historically, and up until some time ago, um, when muscle cells were being, so when muscle stem cells are being used to generate myotubes in vitro, then the first part of division, first few days, when you want multiple cells. We want a lot of cell division to happen. That's in serum, and then serum was removed. So again, what you'll do if you want to get cells from, if you want to culture muscle in vitro, then you need to have a source. That source, again, you can obviously, again, there are ways in which you can buy muscle stem cells, but again, initial source was still there was this muscle tissue that was taken. So again, it was normally it's again you want a source where you have a lot of cells. If you take it from a place where the concentration of these stem cells is low, then you will have to take a larger part of the tissue. So just the same rationale behind using the dorsal root ganglion for your neurons is that you have a lot of neurons there. So the hind leg of mice and rat, that's what's generally used. That is minced. Minced means it's broken down into very small pieces and then it's digested. Digestion is just another name for your dissociation. Okay. And so in that case, you want to sort out your muscle stem cells. These are your mature cells. You don't need this. So you are going to be interested in your muscle stem cells. And then again, so what this bottom step here is telling you is that from your iPSCs, which are pluripotent stem cells, you can differentiate into something called muscle precursor cells. Again, precursor is generally a term which is just ready to become your muscle now. So it's going to be somewhere around your myoblasts, but yeah, that's the thing. So in your once you have your so but we'll mostly be focusing on this first bit here. So yeah, so once you have your muscle cells, muscle stem cells sorted out, purified out of this collection which had multiple cell types, again, you could have used density centrifugation, you could use immunopanning, you could use FACS for this. Once that is done, then you go through the first part is division, the second half of the protocol is differentiation, and then you have it. So again, this is just one diagram just for your, just like an example. So you see on the x-axis, these are different. Again, we did not explicitly talk about that, but what substrate do you grow your muscle cells on? So this was this paper which just talked about, whether you grow them without a coating, you grow on BSA, on laminin, on collagen, on fibronectin. What's the growth? So just to check what substrate is supporting the best. And then the CPK activity, which is creatine phosphokinase activity, that is indicating how much muscle has been differentiated, how much final muscle have you got. So you can see here, so the black line is telling you about when you grow it when it's finally being grown in a serum free media versus when it's grown in media that has horse serum. So the final thing, remember the final thing, serum is no longer needed, right? So you can see in all these graphs, the black line is either equivalent or taller than the white line, right? So that's telling you that serum-free media for the final bit is supporting muscle differentiation better. And out of all of these different coatings, so again, this was if I remember correctly, this was the C2, C12 cell line that they were checking. Yeah, this skeletal muscle cell line. This is what they were checking. And in this case, laminin is working the best. And then collagen, both subtypes of collagen. Remember, we talked about collagen can have multiple subtypes because it is like, yeah, depending on how the helix is being formed. So both the collagen subtypes are the next best. Okay. And then, so again, just some diagrammatic things to tell you. So what we just talked about, you have your skeletal muscle that you generally take from the hind limbs. So that's extracting the muscle, and then you want enzymatic mechanical dissociation. Immunofluorescent staining is done. Muscle stem cells are isolated from FACS. So again, whatever be your X and Y axis, through prior knowledge, you know that, okay, this sort of a population is generally very, very pure in muscle stem cells. So you isolate that, and then you can see there's an approximate time that's given. So you see here, dissection was just given 30 minutes, but then your dissociation and your staining takes some time. So that's given 4 hours. And then fact sorting, because you have to prepare yourself a little bit for fact sorting. You need to go and collect them, and all of that can take a few hours. And then finally, depending on whatever is your downstream application, in case you want to do some cell culture, you want to do some gene expression analysis, you now have a pure muscle stem cell culture, do you want to put it back in the organism, in it's the same or the different organism? Do you want to make some changes in those cells, like we discussed, some transgenic things done and so on? So yeah, so in this case, I was just checking online. So lysine, collagen, and we saw here laminin. These are supporting muscle cell growth, but again, depends. Different. You can have different subtypes. Depends on, like in this case, this is a different cell line. You can use primary cells can have their different preference and so on. Um, One more, so this is where we were at in talking about how to grow muscle cells. Then we talked about that once you grow something, you also want to check if it's grown perfectly well. Phenotype, we've seen in the past. Again, you can do some fluorescence staining and you can look at it under the microscope. Right? Okay, is it forming those tubular structures? Is it aligned well? Do you see these multiple nuclei and all of that? One thing that is often like in case of neurons, we saw that there's a possibility of glial cells contamination because there are a lot of glial cells around nucleus. In case of muscle cells, there are a lot of fibroblasts that are around muscles. So things like this, you need to ensure that your fibroblasts are not coming along when you are purifying your culture. Then what you want to check in terms of their functionality, in terms, so once you've seen that, okay, they look okay, you don't have any contamination, they look like muscle cells only, you want to see if they're working, if they're functional. So two things that you want to check is a calcium spike. Again, we're not going into too much details of how you do this, but that's one thing you want to check. uh, in addition to checking the spike itself, you can also check if there are things which ensure that the calcium spike is produced and what respond to the calcium spike. So there are some of these receptors which are called rhinodone receptor and dihydropyridine receptors. So are these receptors present? Again, you can do some immunofluorescence. Your antibodies can be against these receptors. And if you see that the antibody is showing up, then you know that, okay, at least the receptors are there. Then the next step should be to check if the calcium spike is being produced or not. And then you check the contractility. So the way a muscle fiber, within the muscle fiber, there are two cytoskeletal proteins. One is myosin and the other is actin. So what you can see here is that there's some sort of an alternating layer. So this top layer is your actin, then it's myosin, then actin, then myosin, and so on. And they are arranged in a zigzag way. So you can see here that where there's myosin, there is not actin here in the center. And here, if there's actin, then in the center, there's no myosin. So it's a bit zigzag arranged. That is when it's completely, that's like the medial position. And then there are these other things on the myosin, which are called the myosin heads. Now, in response to some signals, what can happen is if this, let me just take this. Now, if this is pushed here, this actin, and this actin is pushed here, likewise, this is pushed here and this is pushed here, then your muscle is contracted. It's brought, it comes together. Okay, versus if it's the other way, then it's extending. There's another very nice video on this sliding filament thing. I forgot to add the link to this video. I'll add that in the PDF, so you can watch that also if you want. But there's this very nice video which is not just on sliding filament, but everything about muscle, assembly and all of that also, if I remember correctly. I would highly recommend you watch this. I don't know if you would want to watch it right now. Let me know. We can watch it after the class. Or anyway, the link will be with you so you can take a look at it later also. All right. So so again, you start with muscle. You start with the muscle itself. You make a single cell suspension. You label what you want. You culture. You sort out, and that's a pure stem cell culture, stem cell solution now. And then whatever you want to do, if you want to culture them, then of course now we've got not just the serum, but now we have defined media also that you can use directly to for the first phase, for the second phase. And once it's grown, you check if it's working fine, if it looks okay, if it works okay. And then of course, why would you want to grow a muscle culture? Like we saw a use case like in like a case study for neurons that you want to study some disorder. So you take um, healthy neurons and diseased neurons, and you're looking at different drugs on that. Likewise, you can also take muscles from a mice which is healthy or a mice which has some disease. You culture them, you test drugs, or the other way around, you take a healthy muscle and you're introducing some injury onto it, and then you're testing drugs onto it again, right? So again, there's a lot of applications. Please let me know if you have some questions up until this point about this, or if there's anything you want to tell, some example, if you have worked in this field, or if you've come across any study, anything interesting that you just want to talk about, then let me know at any point of time, now or later. Okay. So do I still have you with me? Okay. Yeah. Let me know if everything's okay. Um, yeah, I don't think I see a thumbs up if the screen is minimized. But yeah, okay. That's all good. So next we go on to cardiomyocyte culture. So yeah, now in this case, again, the principle is quite similar. You're talking about you can, again, if you just want like a pure culture, it's easier to do with an embryonic tissue. So far we've been talking about embryonic or very early neonatal tissues. So again, mice heart is dissected out. When it's being dissociated, when you're chopping up into smaller pieces and dissociating mechanically or enzymatically, it has to be in some medium. There has to be some liquid there. So the L15 medium is used. It's one medium. B27 and glutamax, which were also used for neurons, that's also going to be supporting heart cell growth. It's dissociated. Again, pelleting is it's a very common step because you do want all the not so initially they're going to be in a bigger volume, and then if you want to reduce the volume, you allow the cells to settle down, which is very gentle spinning. So between 300 to 500 RPM is generally what's recommended for 3 to 5 minutes. A little bit up and down depending on how sensitive the cells are. You remove whatever media you have. You now have very often what people also do is that you can literally filter your cells. So what you can do is, suppose you have this tube and then you have another tube. So this is where your cell suspension is. So what you can do is this is some sort of it's called a cell strainer. Oopsies. Mind my writing here. So generally this is, you do get in different sizes, but let's say you have something like a 40 micron cell strainer. So it's like a mesh, a very, very fine mesh. You add your cells and they'll go dropwise into, and they'll get collected here. So what's going to happen is, if you did not have perfectly single cells, but you're like, okay, I don't want to dissociate anymore, otherwise I risk breaking down my actually good single cells. So it's like this. So if you have 100 cells to begin with, maybe you got 60% single cells and 40% were in chunks, in some, clumps, or yeah, they were not single cells. So what do you do? Do you try to break down and pipette more? Because then, okay, while you may be able to break some more of your chunks, but you risk stressing out your single cells. Or do you do something like this? Do you strain out? So you do get 60% of the cells, but you completely lose that 40%. So again, it depends how precious that material is. I mean, obviously, all material is precious, but how little cells do you have? Do you want to pipette? Do you want to dissociate with trypsin or with any enzyme? Do you want to dissociate it more? And then, but also, you risk killing some of your cells. Or do you do this, which is gentle, which is really gentle, but of course you'll lose something. You'll lose all the clumps. Sometimes you'll also lose some um, healthy single cells also, because they'll just stick there. But yeah, this is very sterile. This is a one-time, very small strainer. So this is a very, it's often used, and it's definitely recommended before fax. So cell strainer, generally it's always recommended, because like we talked about, this nozzle here, this is a very fine nozzle. 70 microns, 100 microns, that's the kind of size. Your microns is micrometers, so mu m. So this will get clogged if you have cells which are bigger. If you add chunks into it, it's just going to block your nozzle. So you don't want that. So definitely before fax sorting, cell has to be strained. They have to be filtered. They have to be passed through that filter. Okay. Alrighty. So yeah, so that's the how the cell strainer works. And then substrate that allows hard cells to grow well. Laminin, fibronectin. Again, some of these are newer names, but you don't necessarily need to remember this. Uh, Plating media, we just add some sodium bicarbonate to it so that it's buffered when it's in 5% CO2 incubator. Plating density, again, very similar. They are going to divide and grow, but you don't want to over plate your cells. You don't want to oversaturate your well or your plate, because all of these cells, they like to grow as a single layer, but you don't want them to be too separated out also, because they prefer to grow together. You want to allow some migration. There has to be space, but you don't have to have too much also, too much empty space also. And this is what it's going to look like a little bit, beating in some sort of a wave. Okay, they're all connected. You can see there's a lot of connections, but they're all um, beating in as one single entity. All right, so that's that. And yeah, so again, what you want to check is some expression of some things. These also have some, because this is a muscle again, so some myosin is expressed there. You can look at that particular subtype of myosin, some gap junctions, because they all get connected, so it's a lot of gap junctions are there. Of course, you can look at these waves in terms of their functionality, calcium wave, the actual phenotype of it beating, and all of that. So that's how you can check if your cardiomyocyte culture is um, working. And oopsies, sorry. Yeah. So yeah, so that's about it from the purpose of the discussion. Do you have any questions until now? So what we'll do next is I just have some, I think around 10 questions to quickly revise what we saw this week. And here I would really want if you could quickly put up your answers in the chat. Uh, just make it a little bit more interactive. And these questions are going to be very similar to your assignment questions. Not the same, but you get an idea. And then, of course, for those of you who've registered for the exam, it should help you with that as well. Okay. Alrighty. So, which of these techniques can be used to separate a population of two cell types? from a mixture. Immunopanning, fluorescence-assisted cell sorting, both A and B, none of the above. So yeah, so both immunopanning and FACS can be used. All right. Sorry. Let's put this here. So in DASH, there are two types of motor neurons. One are small in size called that, the other are large. Okay, so we did not explicitly talk about this here. Okay, so let's go back to that figure actually. I think that's what I wanted to tell in that figure, and then I got distracted. So if you see here, on the back side is where you're getting your sensory neurons. Okay, so the back or the dorsal side is where the sensory fiber, sensory neuron, sensory nerve, all of that is coming in. And then on the output side, so the thing is that your spinal cord is taking input in the form of some senses, and the output is an effect that's a movement. It's the output is that it's telling you, okay, move away, move this side, and so on. So that's a motor neuron function. So the output, which is from the ventral side, that's the motor neuron. And then, if I remember correctly, the alpha subtype is larger than the gamma subtype. So motor neurons are from the ventral side. That's the ventral horn is where they are coming from. So this is where your motor neuron are coming from. So what I've written here is, so the spinal cord, if you just say that the spinal cord is composed of your dorsal ventral horn, that's the CNS. So everything that's inside, the part of these sensory neurons and motor neurons, part of which that is inside these horns will be called as a part of the spinal cord, and the part that is outside is called a part of your PNS. But yeah, so again, coming back to this question now. So in what are your motor neurons? Would you like to stay with the same answer, or would you want to change it now? Okay, yes. So what do you think the answer would be? So the motor neurons are now in the ventral horn, right? And the larger ones are actually the ones small in size are gamma, and the larger ones are alpha. So I think the answer should be 4, but yeah, I think the size, if I remember from the lecture, the smaller ones were gamma. So it should be 4, but yes, a very good attempt. So thank you for that. Okay. Then we move on to the next question. So in the right spinal cord at E14, only motor neurons have this particular marker like we saw. So what can you do with this information? Can you isolate the cells when it's in a mixed culture with other cells from the spinal cord? With this information, can you test the electrical activity? Can you digest these cells, or can you not do anything with this information? So once you know that at a particular stage, a particular subtype of cell is expressing a particular marker, then with that information, you can isolate the cells if they are mixed with something else. Okay. Now I don't have any options for this. So what is a protein molecule that recognizes something on the cell surface? Your options are antibody and antigen. Which comes where? So what recognizes what? So would any of you like to attempt this? So generally it's like this. So this is your antibody, and then that's an antigen, and this is where your detection is. So that's your antigen. Oopsies, sorry. So that's your God, it's very difficult to type like this. Okay, so that's your antibody. Okay, yes. So, okay, so I mean, honestly speaking, it is a two-way interaction. But the way we generally, yes, the way we generally talk about this is that the antibody is recognizing an antigen. So this is the antigen. Maybe this is something on your cell surface. Maybe this is some secreted molecule, whatever it is. And this part, which is a part of the antigen that the antibody recognizes, you might hear this term somewhere. It's called the epitope. So the idea is that an antigen, it's a big molecule, right? An antigen, let's say it is a protein which has let's say 500 amino acids. Okay. So now the first 10 amino acids may be recognized by something. The middle 10 may be recognized by something. Some 10 at the end could be recognized by something else. So you can actually have different epitopes on this antigen, and each of them can be recognized by a different antibody. So the way this goes, I mean, both recognize, like either is recognizing the other, but the way it's generally said is that what you have on the cell surface is an antigen, and against that, the detecting part is the antibody. Okay. So yeah, so antibody recognizes the antigen. All right. Good try again. So what happens to myoblasts during myogenesis? Again, myoblast is that once your muscle stem cells, once your satellite cells have activated, once they have differentiated, we call them myoblasts. And myogenesis is making genesis of myo, so muscle, making muscle development. So yes, so they fuse together to form multinucleated myotubes. That's right. Then, what is the main function of satellite cells in adult skeletal muscle? So, why do you have these? So, satellite cells are your stem cells, right? Why do you have stem cells in an adult tissue? It's already grown, right? You don't need to grow it again, do you? Yes. So, you don't need to repair and regenerate if there are damaged muscle fibers. So, it's not a perfect thing. So you can hear things like some scar tissue, especially that's more the case in cardiac muscles, because it's not a great perfectly it's not a perfect process. But yes, the stem cells are there for when there is a need, things can be repaired. All right, good. What happens to the myoblast when the serum is withdrawn? So initially, you have the serum, then when the myoblast is proliferating. But what happens is when the serum is withdrawn? So we talked about the first case where there was a lot of proliferation, a lot of cell division, and then we talked about differentiation and fusing. So yeah, so the proliferation decreased. So here it means proliferation, and then your cells began to fuse. Okay, so proliferation does not increase when serum is withdrawn. Proliferation does not stay the same. And apoptosis is cell death. It is programmed cell death. So cells are not dying when when serum is removed. They just stop dividing. So their division decreases, but they do something else, which is fusing. Okay. Which cell types tend to overgrow in cardiomyocytes in primary cultures if it's not controlled? We did not explicitly talk about cardiomyocytes, but it's the same case. Is it smooth muscle cells? I thought it should be fibroblasts. Is it smooth muscle cells? So I was thinking, like in case of skeletal muscles, it's the fibroblasts which can overgrow. I think it should be fibroblasts, but I don't know. Is it smooth muscle cells? No. In my opinion, it's fibroblasts, but let me double check. But yeah, I think it's going to be fibroblasts. Yeah, this is cardiomyocytes, you're right. But even in cardiomyocytes, I think it's going to be the fibroblasts which can overtake. But let me confirm. Okay, I'll just double check this. Yeah, thanks for answering. Yeah, all right. And then trypsin is used to do something, and it does that by digesting the extracellular matrix as it is a. So, does trypsin lift the cells off? Is it used to attach? It's a weird sentence framing here, but yeah. So, trypsin is used to lift cells off the surface because it is a proteolytic enzyme. All right, great. Option two. And then the final question. So, I will just put this up here for a second. So, read the statement about animal origin free medium. So when we are not talking about a serum, but instead we are talking about a defined medium, which statements are true? That so this medium, let's say we talked about neurobasal and things like that. So that's not manufactured using any raw material that's derived from animals. That could be human tissue, cell, body fluid. No animal derived material is used as a starting point or at the primary level manufacturing process. Is the statement true or not? Animal origin free medium. Also, the components used to manufacture the raw material, those ingredients are also not derived from anything. So, the primary level, the secondary level, right? The ingredients and components may be produced from plants, bacteria, yeast, or cell free systems from them, or they may be produced by actual synthesis in the lab. And the fourth statement is they may contain recombinant animal or non animal proteins, provided that they are expressed in plants or microorganisms. So, which of these statements do you think is true when we talk about animal origin free medium? So, if you go and look at how neurobasal A was made, or any other medium for that matter, which is not which is a defined medium, which statements do you think would be applicable? So option one and two, yes, you're right. Option one and two are applicable. So it's not manufactured using any raw material at the primary or the secondary level. But also, the thing is that it still has certain things. So where are they coming from? So it's yeah, so they could be produced in plants, bacteria, or yeast. And these could be recombinant materials. Which are expressed in either of these organisms. So these are animal origin free, but these things are not animals. So they could be directly produced in that, or they could be like transgenically expressed in these things. So all the four statements are true. Okay. Alrighty. So with that, we've come to the end of this discussion. I would like to quickly walk through the week 6 assignment, and we'll just come to one question that somebody had raised a question about in the question answer forum. So this should just take a couple of minutes. So, what type of cells are responsible for myelination in the peripheral nervous system? And that's the Schwann cells. They are the oligodendrocyte sort of contemporary because they themselves just wrap around and secrete the MBP. Which animal model is commonly used to isolate the dorsal root ganglion? So it's both, it's both, but rat more than mice. I mean, this was explicitly talked about in the lecture, so I hope there's no confusion about this. But yes, of course, you could use the mice also. Why is trypsin inhibitor added when you do an enzymatic dissociation of not just dorsal root ganglion, but anything? So trypsin will keep on working, and once it has digested the cell-cell contacts, it can also affect the cell, the membrane proteins because it's a protease. Okay, so it can chop any protein inside. It'll start off with the proteins which are exposed, but it can do damage. So these protocols would often tell you that, do it for 5 minutes with this concentration, or with a different concentration, it's going to be for 10 minutes. and then you need to stop the reaction. For different enzymes, the reaction stopping can be enzymatically. So you are adding an enzyme here itself. This inhibitor would itself be some sort of an enzyme. Or in some cases, depending on how sensitive your sample is, you may put it at a different temperature, whether a higher temperature or lower temperature. So higher temperature proteins will, they'll unfold, so they're not going to work. At a lower temperature, enzymes will lose their efficiency. So the cells and all will stay okay. The cells are happy at 4 degrees also. They'll just be quiet, but your enzyme will no longer work. So cold, high temperatures, they're both used in different cases. So anyway, trypsin reaction has to be stopped so that there's no damage to cells. At what stage would it be ideal to introduce Schwann cells into the culture of neurons? So you don't want Schwann cells to overtake. So initially, you just have neurons, you allow them a few days to extend their processes to grow, and then you add Schwann cells. So of course, you want to grow Schwann cells along so that the neurons get myelinated, but you don't want them to overgrow. All right. What protein do they secrete? That's the myelin basic protein. That's the myelin sheath. That forms the myelin sheath. Um, when which of the cells would divide actively in cultures? So that's going to be Schwann cells. Neurons are mostly non-dividing. They can have processes increasing, so their axons and dendrites can be moving here and there, but the number of cells are not going to increase. Uh, Postnatal day that is used for rats to isolate Schwann cells. It's very early on, so postnatal 1 to 4. What is the major challenge when you are culturing a non-dividing cell, which is neuron, with a dividing Schwann cell? Yeah, they're just outnumbered. We just saw that again. Which substrate is commonly used to enzymatically dissociate the dorsal root ganglion? So while in the lectures, trypsin was mentioned explicitly, but collagenase is also used in real settings. So if any of you have put up collagenase as the answer, which I think somebody did in the, if you go to the Q&A page, somebody has asked this question. We've not heard back yet. Okay. This thing, collagenase. So you could answer back. You can just reply onto the same thread. But yeah, so again, these are both proteases. So they would do a good job. It's just that collagenase are going to be more specific to collagen. But trypsin will also do the job. So it's not wrong. But yeah, if any of you have ticked collagenase as the answer, then you can just put it up there. And yeah, neurons, they are your non-dividing cells. All right, then. So that's your week 6 assignment. And yeah, I think we've covered everything. So we just connected some dots from last week. Facts was discussed in detail. It's very, it really is a fancy technique, but it's very powerful, and it's just a few things that need to be taken care of. Especially, you have to ensure that whatever marker you're using can be labeled with an antibody, with a fluorescent antibody, and that marker is specific to the cells you are interested in. Then we saw a use case, and then we talk about muscle cells in this week, which is your skeletal muscles and your heart muscle cells. So since in both of these cases, we are sort of working from a stem cell to a differentiation paradigm. So it's important to check their phenotype and their functionality, because there's going to be a difference because of there's an in vitro setup instead of an in vivo setup, and you're doing that differentiation also in vitro. Skeletal muscles, that's also skeletal muscle, and then cardiac muscles. All right, then. So that's it for this week. Thank you for joining. The recording is going to be uploaded soon. I will put up this PowerPoint as well. There was one thing, so I'll just confirm this answer here. I think I can just, yeah, can I just Google it right now so that I won't forget it later? Do we see something in here? Not interested. Yeah, I think it's mostly fibroblasts, cardiac fibroblasts. Yeah. So I think the answer stays the same then, fibroblasts. But yeah, but thanks for answering it anyway. All right. So the deadline for this week's assignment is yes. All right. Thank you. So yeah. So the deadline for this week's assignment is the after tomorrow, Wednesday, assignment 7, and then we will meet again next week for the last lecture, last live session for this course. Um, so I'll just let you know that after this, we would not have any other lecture. There's no revision session for this course. I mean, if you've joined some other NPTEL classes, you might have it for some of those. But yeah, so for the next week's assignment, then we won't have any session where we'll explicitly discuss that, but you'll anyway have your Q&A forum available. So use that for for the assignment of next week. But yeah, anyway, I'll see you all again on next Monday, 13th. And thank you for joining. I'll stop sharing my screen now. Yeah. Bye, everybody. Good night.